And we are here today to have a hearing entitled Rare Diseases Expediting Treatment for Patients. And I'll begin with an opening statement before we introduce the panel. Through our hearing today, I intend to examine regulatory barriers to entry for drugs that treat rare diseases, also known as orphan drugs. The FDA defines orphan diseases as those with fewer than 200,000 patients. There are over 7,000 conditions that science has identified affecting nearly 30 million patients just in the U.S. Unfortunately, about 90% of these are serious or life-threatening conditions, and most have no FDA-approved treatment. The standard for approval of orphan drugs is similar, if not the same, as those for more common conditions. It can take upwards of a decade sometimes for a drug to make it from the bench to bedside, and costs can be upwards of $2 billion. For terminal patients waiting for the cure that may never come, or one that comes too late, is simply not an option. Therefore, families and patients often resort to importation of drugs approved overseas, off-label use, or may turn to dangerous chemical-grade products. As a physician, I understand how rare diseases affect the body and manifest themselves through devastating and debilitating symptoms. Even in my family alone, I have a nephew who has neurofibromatosis too, and we've had to deal with a disease that's not very common and many people have not seen, even many physicians have not seen. As a parent, I can only imagine, though, the overwhelming desperation and hopelessness of being confronted with a fatal diagnosis and being told to simply enjoy the remaining time that their child has left. To the extent that government can improve efficiencies to speed access to treatments for patients and families facing such a situation, such action should be taken without delay. Despite providing the FDA with numerous tools to expedite approvals and continually increasing resources, review times have not decreased. Over the life of the current user fee agreement, the FDA will take in more than $4 billion in user fees from the drug industry, amounting to over 70% of the agency's review budget for these products. Since the beginning of my time in the Senate, I have worked to ensure that the FDA has had the authority to accept data from foreign countries. Yet despite these efforts, duplicative trials and testing are still required in order to bring most drugs to market. In addition, drugs approved in Europe are not harmonized with the FDA's process, causing problems in an increasingly globalized scientific community. I hope we can work together today and in the future to find solutions that work to expedite treatment for these patients. With that, I'd like to recognize the ranking member, Senator Casey.
Thank you, Chairman Paul, for calling this important hearing. I want to thank the witnesses for being here and thank the chairman of our committee, Senator Alexander, for his presence here. I wanted to apologize in advance. I have to leave in about 20 minutes. This hearing was scheduled at a time when I had a conflict. I wish that hadn't happened, but it did. So I have to leave early. I'll be submitting questions in writing for the witnesses, and I look forward to your responses. But we're here today to talk about a critically important issue, the development of drugs to treat rare diseases. 35 years ago, Congress passed the landmark Orphan Drug Act. This legislation represented the first concerted federal effort to incentivize the development of new treatments specifically for rare diseases. The Orphan Drug Act established grants to assist in the development of new orphan drugs. It established the Orphan Drug Tax Credit to further offset the research and development cost, and provided a longer period of exclusivity, a full 7 years, for orphan drugs. As a result of the act, over 600 orphan drugs have been approved and many more in development. This compares to just 34 orphan drugs approved in the 15 years before the act was in effect. In recent years, Congress has taken a number of additional steps to streamline and speed up the development of new therapies in rare diseases, or for rare diseases. The first I'll mention is one that I'm quite proud of because I developed this policy with Senator Isaacson. This incentive is the Pediatric Rare Disease Priority Review Voucher Program, which rewards the development of a drug for a rare pediatric disease with a voucher that can be used to give another drug priority review by the FDA, cutting the review time from 8 months to 6 months. Companies can keep the voucher to use it for another drug in their portfolio, and they can sell it and reinvest it in their own research. I'm grateful for the work of Senator Isaacson on this with me. Since the Pediatric Rare Disease Priority Review Voucher Program was established in 2012, 13 vouchers have been awarded for new drugs to treat rare pediatric diseases, for diseases ranging from cancer to a genetic cause of blindness, to the first marijuana-derived cannabinoid drug to treat two rare forms of epilepsy. The FDA has a number, I should say, of other tools at its disposal to aid in the development and approval of drugs for rare diseases, several of which have been enacted in the last 2 years. The same law that extended and strengthened the Pediatric Priority Review Voucher Program, the 21st Century Cures Act, also required FDA to develop guidance on how to use adaptive trial designs and Bayesian method for clinical trials. FDA published the draft version of that guidance last month. The same day, FDA also published guidance on the so-called master protocols, which can allow a single clinical trial to evaluate multiple drug candidates, multiple disease types, and more than one patient population under the same clinical trial structure, potentially reducing the time and cost of generating the necessary data for approval. One of the common points we hear from the patient community and the companies working to develop new drugs is that patients are, and understandably so, desperate for access to new drugs. That is hard to enroll them in clinical trials. Congress has given the FDA specific authority in the FDA Reauthorization Act, I should say, to consider ways to design clinical trials that can incorporate data from expanded access use of investigational drugs. The FDA is also learning from the adaptive trial designs employed during the Ebola outbreak in 2014 and using those lessons to inform other clinical trials where the standard randomized, I should say, double-blinded placebo trial is not possible or is not ethical. The agency has noted that these new study designs are relevant to the growing field of gene therapy and thus also to the rare disease community where so many of the diseases are genetic in origin. I look forward to continuing to work with the patient community, other stakeholders, and members of the committee to advance drug development for rare diseases while continuing to ensure patients can trust their drugs and trust that those drugs are both safe and effective. Sure. Thank you very much.
I'd like to recognize the chairman, Senator Alexander.
Thank you, Mr. Chairman. I'd like to thank the witnesses for coming. We look forward to this. Senator Paul and Senator Casey have been real leaders in our efforts to put a spotlight on rare disease. I want to thank Senator Paul for the hearing. I want to thank Senator Casey for his work over the years and his contributions to the 21st Century Cures Act. As with Senator Paul, I look forward to the testimony. We have a vote. I'm managing the opioids bill on the floor when the vote's at 3:15, so I'll have to leave a little before that. But I look forward to the hearing. I salute Senators Paul and Casey for their focus on such an important topic.
Well, thank you both for coming. As you'll see, if you haven't been to a hearing before, we have multiple hearings going on everywhere around, as well as votes on the floor. So sometimes you see people come and go, but I do thank Senator Casey and Senator Alexander for coming. We're going to start with testimony from Mark Dant, who is the chair of the Every Life Foundation for Rare Diseases, and also from the great Commonwealth of Kentucky.
Chairman Paul, Chairman Alexander, Ranking Member Casey, and distinguished members of the Help Committee. I am Mark Dant. I am the chairman of the board of the Every Life Foundation for Rare Diseases and the parent of a child with a rare disease. the Every Life Foundation is a science-based advocacy organization that works to bring life-saving treatments to the 30 million Americans with rare diseases. We represent the 1 in 10 Americans affected by more than 7,000 known rare diseases, with 50% of the rare diseases patients being children, many of whom will not live to see their 5th birthday. It is imperative that we significantly increase the number of FDA-approved rare disease treatments now. When my son Ryan was diagnosed at 3 years old with MPS I, a rare lysosomal storage disorder, the cells in Ryan's body lacked a crucial enzyme that they need to break down sugar. The hospital told us that children with MPS I rarely, if ever, lived past their teens, as there was no treatment for his disorder. Because MPS I was so rare, affecting only a few thousand children around the world, drug companies were not interested in funding the research. My wife, Jean, and I were told there would be little other that we could do for Ryan other than take him home and love him for as long as he lived. I refused to accept the recommendations. I spent all my spare time trying to understand Ryan's condition, and after a year, my wife, Jean, and I founded the Ryan Foundation to raise money to find a treatment for MPS in time for our son, Ryan. Our first fundraiser was a bake sale that netted $342. Eventually, I learned of a pediatric researcher who was working to find treatments for children with MPS, Dr. Emil Kakis at Harbor UCLA. It was 11 years after the passage of the Orphan Drug Act, yet Dr. Kakis had no funding for research. Over the next several years, the all-volunteer Ryan Foundation raised more than $1 million for Dr. Kakis' work, which culminated in a new drug therapy. This therapy would never have come to fruition without the formation of a small biotech company, which pulled Ryan's drug through the pipeline in time to help him survive. Rare disease absolutely needs biotech partners. Family organizations like my own simply do not have the capital necessary to bring treatments to approval. In 2003, the FDA approved Aldurazyme for the treatment of MPS I, 5 years after Ryan and 9 other children began the trial at UCLA. Enzyme replacement therapy later turned out to be instrumental in treating several other previously untreated and devastating disorders, proving again that biotech involvement in one disorder leads to not one, but countless more disease treatments. Ryan is now 30 years old, a graduate of the University of Louisville, and is the longest treated person with MPS I in the world. Unfortunately, Ryan's story is the exception. We are now 35 years since the Orphan Drug Act was signed into law, yet fewer than 400 of the 7,000 plus known rare diseases have FDA-approved treatments. We call on Congress to close the innovation gap for the 95% of rare diseases that have no treatment by incentivizing companies to repurpose already approved drugs for rare disease. Many patients are using drugs off-label, including my own son, Ryan. Drugs used off-label do not have the appropriate safety, efficacy, and dosing information. They also lack coverage for the cost of the drugs, as many insurers will not pay for off-label use. The bipartisan Open Act, S-1509, introduced by Senators Hatch and Menendez, is a patient-driven solution supported by more than 300 organizations, including my own. Modeled after the bipartisan Best Pharmaceuticals for Children Act, which resulted in over 600 labeling changes, the Open Act has the potential to double the number of FDA-approved therapies for rare disease patients and at a lower average cost than current rare disease treatments. I urge Congress pass the Open Act before the end of this year. In addition, we call on Congress to do the following steps. Fund a center of excellence for rare diseases. Allow innovators to use novel biomarkers for rare disease research. Shorten the 7 years it takes to obtain an accurate diagnosis. Pass the Newborn Screening Saves Lives Act, and pass legislation to ensure coverage of genomic sequencing. In conclusion, I ask all of you gathered here today, Republicans, Democrats, Independents, to please put your policies and politics aside and join the RARE Party. The FDA is not our enemy. Biotech companies are not our enemy. Our enemies are the rare diseases that steal livelihoods, mobility, vision, minds, and in the worst cases, lives. All of us, patients, policymakers, innovators, must work together to speed the development of and access to safe and effective treatments for the rare disease patients across the nation. Thank you.
Well put. Thank you for your testimony. Our next witness is Dr. Mark Patterson, who is a professor of neurology, pediatrics, and medical genetics at Mayo Clinic.
Chairman Alexander, Chairman Paul, Ranking Member Casey, and members of the committee, I wish to thank you for the opportunity to testify before you today, for your interest in this important topic and the work that you've already done. I'm honoured to have this opportunity to advocate on behalf of children and families afflicted by rare diseases. My name is Mark Patterson. I'm a paediatric neurologist, and I currently serve as a professor of neurology, pediatrics, and medical genetics at Mayo Clinic in Rochester, Minnesota. I completed my fellowship training in rare diseases at the National Institutes of Health some 3 decades ago, and I've dedicated my career to children and families with rare disorders since that time. I've cared for many hundreds of children and families, and as you can gather from the testimony of the previous speaker, and understand why I admire the courage, the creativity, and resilience of these extraordinary families who are my personal heroes. Congress has recognized the plight of people with rare diseases for more than a generation to provide needed incentives for researchers to devote resources to investigate and develop therapies for rare diseases and strengthen interest in rare diseases at the National Institutes of Health and to encourage the Food and Drug Administration. These acts of Congress are widely regarded as having been highly successful in stimulating the interest of industry in developing orphan drugs, and I thank Congress for that work and the work that you have done in furthering those ends. I'd like to emphasize the fact that advances in diagnostic techniques, particularly the next generation sequencing of DNA, have led to the rapid expansion of the number of recognized rare and ultra-rare diseases, by which I mean diseases that affect fewer than 2,000 individuals, sometimes as few as 10 or 20. Collectively, these diseases affect a very significant proportion of the population, as you've heard, yet few of them have approved therapies. Moreover, the increasing use of next-generation sequencing means that disorders which we currently think of as common diseases will likely prove to be families of rare disorders in the future. So I think there's considerable urgency in finding better ways to develop treatments more rapidly. As you already gather from Mark Dant's testimony, every family's story is unique, but there are certain common themes. The initial symptoms of rare and ultra-rare diseases are often mistaken for those of more common disorders. Families will travel from physician to physician, from medical center to medical center, enduring extensive, expensive, and sometimes invasive investigations before the correct diagnosis is eventually made. And often that diagnosis is delayed by years. By this time, the opportunity for early and effective intervention has often passed. The patient and their families then enter a new and equally frustrating stage. They face caregivers and others who are unfamiliar, sometimes even with the name, let alone with the burdens of such a diagnosis, and they have to deal with a bureaucracy which is largely designed to care for adults with common diseases, not children and young adults who have progressive disorders. Often, families are told inappropriately that nothing can be done for their child. The process of developing any new treatments, specifically pharmaceutical therapies, is long and complex. Typically, this process requires large numbers of subjects who will ultimately participate, as we've heard, in randomized, double-blind, placebo-controlled clinical trials. But this pathway is inappropriate and has many barriers for rare and ultra-rare diseases. As we've already heard, there's a small potential pool of participants. Not all individuals are suitable candidates or are willing to participate, and there's wide individual variation from individual to individual in terms of the symptoms, the age of onset, and the rate at which the disease progresses. All of this makes the assembly of well-matched cohorts of patients for controlled trials well nigh impossible. Another challenge is how to measure the effects of drugs in rare diseases, because traditional measures are usually lacking. While there have been many attempts to address these deficiencies and there's been progress at the FDA and the NIH, there are still fundamental challenges remaining. The bottom line is that we need more effective methods and pathways for drug approval for rare and ultra-rare diseases. I suggest that Congress and the administration consider a variety of approaches, a few of which I describe in my written testimony and which I will highlight now. And I have 3 themes. One is trial design, which Senator Casey has already addressed. The second is the use of data from outside studies. And finally, I'd like to address the issue of registries. First of all, I think that we should ensure that the FDA will accept alternative study designs, including adaptive trial designs and Bayesian trial designs, which have been mentioned, and ensure that patients will have access to the study drug to encourage participation. Secondly, the FDA should be required to accept the results of well-conducted clinical trials supervised by national regulatory agencies outside the United States or by such agencies acting in concert with the FDA. Finally, we should require the FDA to work with family groups, academic medicine, industry, and other international regulatory agencies to develop disease registries which contain secure patient and parent-entered data which can be used to enhance understanding of natural history, to develop outcome measures, and to support clinical trials. I thank you for the opportunity to present these suggestions to the subcommittee, and I urge Congress to consider providing regulators with a new, improved set of tools to ensure the translation of scientific and technological advances to safe and effective medicines for the millions of Americans suffering from rare and ultra-rare diseases. I look forward to answering any questions you may have. Thank you.
Thank you, Dr. Patterson, for your testimony and also for your lifelong work in trying to find cures for these awful diseases. Our next witness is Dr. Michael Strupp, who is a professor of neurology at the University of Munich. And when you conclude your testimony, if you want to introduce and show a couple of videos right at the end of your testimony, we'd love to see the videos.
Senator Paul, Senator Casey, Alexander, and Senator Cassidy, thank you for the opportunity to present our suggestions. In my statement, I'm going to focus on 4 topics. First, what is meant by ultra-rare diseases? Secondly, what are the specific problems in such diseases in terms of designing and performing a clinical trial to get approval? Third, what would be the impact of an adopted approval process on research and even investments by pharmaceutical companies? Fourth, and finally, my vision. What does rare and ultra-rare mean? Senator Paul already mentioned that rare diseases are defined by fewer than 200,000 people affected by a certain disease in the US. If you take into consideration a population of 325 million Americans, you will end up with a prevalence of less than 60 in 100,000. There are, however, ultra-rare diseases with a prevalence of less than 100 of that. 2 examples are Niemann-Pick type C, with a prevalence not of 60, but of 0.7 in 100,000. Tay-Sachs, 0.3 in 100,000. This means, for instance, for Washington, D.C., with a population of 650,000 inhabitants, 5 patients with Niemann-Pick type C and just 2 with Tay-Sachs. What about clinical trials in ultra-rare diseases? Designing and performing a clinical trial in such diseases is very challenging for 4 reasons. First, the small number of individuals that can be theoretically recruited. Secondly, patients with these diseases are often severely impaired, are not able or willing to participate. Most patients are children, and for instance, in Niemann-Pick type C, 50% of the children die before the age of 13, which makes recruitment, of course, even more difficult. And finally, the statistical design, which was already mentioned by my colleague. So the conventionally used so-called sample size calculation with a sufficient statistical power can only be theoretically implied in such diseases, but will not be useful if you have, for instance, only 16 patients in the US suffering from a rare gangliosidosis. Nevertheless, we have to perform clinical trials to get an approval in ultra-rare diseases. From my point of view, there are 3 prerequisites which have to be fulfilled for such trials and which can be called the triple D. Disease, drug, design of the trial, in particular in terms of outcome measures and statistical analysis. Disease in terms of 4 aspects. The prevalence should be really low, less than 1 in 100,000. A precise diagnosis has to be made nowadays by genetic testing. The disease should have a high impact in terms of signs, symptoms, but also functioning and quality of life, not only of the patient, but also the caregivers. And there's a high medical need and no other treatment available yet. When it comes to the drugs, there are also 4 aspects. Ideally, a drug should already be approved for a different indication. That means repurposing of drugs. It should have a very good safety profile. The efficacy should be shown in animal studies without a placebo effect, and the mode of action should be known on every level. That means histopathology, electrophysiology, even up to proteomics. And finally, the design of the study and the outcome measures. This should always be clinically meaningful in terms of functioning and quality of life. Imagine a patient is now able to walk or use the hands properly, take the tablets, take the meals by him or herself, as you will see in one of the videos. This is a major improvement. What could be the impact of adjusting the approval measures for pharmaceutical companies and research? We live in a world of commerce, investment, making money, also by pharmaceutical companies. This is important not only for the shareholders, but also for patients. How is this related to the topic? If the approval of drugs for ultra-rare diseases is adapted to the real world and transparent for such diseases, I think these diseases will become commercially more attractive. Companies will invest money because they have a chance that the drug is going to be approved within a reasonable time. What is my personal vision? We live in the century of personalized medicine. And this attitude, I think, should also be transferred to rare diseases so that we have an individualized and harmonized approval process for therapies based on solid science and the safety of drugs. Finally, as a passionate doctor, seeing 2,000 patients per year, a final statement: one should not withhold effective drugs from currently severely affected individuals because of regulatory approval processes which may no longer be justified. And I'm going to show, we have several examples. There's just one patient with Niemann-Pick type C. We have published a case series, which is our approach to find new drugs on 13 patients. And here you see a 14-year-old girl which should perform the so-called 9-hole pack test, which is a standard test to evaluate hand coordination. In a few moments, you will see what happens if this girl is on medication. So this is just a simple case, and that is our approach. So we use a drug which has been approved for other indications, which is definitely safe, and then we start with small case series, knowing the mode of action of the drug. This drug normalizes membrane potential. We complement that by back translational research with this drug, with the Department of Pharmacology in Oxford, in appropriate animal models, showing the same effects with this drug in the dosages, which are per kilogram identical to the patients.
So this drug is approved where?
What?
This drug is approved where?
This drug has been approved since 1957 in France to treat patients with vertigo and dizziness. My background is electrophysiology. I know the mode of action. It normalizes membrane potential. And therefore, I thought it may have an impact on various neurological disorders to bring neurons which are still alive, but which are not working properly, into a normal state. And this we have seen for Tay-Sachs disease. For
Do you know how long that the application process has been going on in the United States for this drug?
So we had July the 17th, an FDA meeting. That was our first meeting with the FDA. We were able to introduce 2 new outcome measures for our trials, and the FDA was quite positive that we can run a phase 2 trial in the US. And we have also got an approval by the European agencies in UK, Spain, and the Netherlands, which were quite enthusiastic, in particular since this drug has an excellent safety profile, it's well tolerated. And by the way, hundreds of patients take the drug, now worldwide, off-label, of course, from the Philippines, India, to Latin America, and also in the US.
Thank you. Since I've already broken the rules and I know Senator Casey has to leave, do you want to interject with a question now before you leave?
Maybe I'll just wait for the... I'll wait for the testimony of the following.
That's fine. Our next testimony will come from Lincoln Sang, a partner at Arnold & Porter in London.
Mr. Chairman, ranking member and distinguished members of the subcommittee, my name is Lincoln Sang. I'm grateful for the opportunity to discuss certain technical and regulatory issues relevant to facilitating research and development approval of new treatments to ensure their timely access by patients with rare diseases. My statement is drawn upon my experience as a medical scientist, a regulator, a former regulator, and now a private legal practitioner. We've heard the dispassionate and very powerful statements from the patient representative and two eminent clinicians with extensive experience in the care and management of patients with rare diseases, as well as expertise in clinical trials for innovative products in these disease settings. I therefore will limit my statement to reinforce what they have already said, but will place greater emphasis on the regulatory policy based on my experience and understanding of the regulatory landscape in the United States as well as in the European Union. There is a greater need now for new methods of diagnosis and treatment for rare diseases. Healthcare delivery is now increasingly focused on planning the patient journey to improve quality and efficiency of clinical management and to alter the focus of care towards the activities most valued by patients. The regulatory landscape may need to reflect more accurately the advances in science and technology and a changing treatment paradigm which is increasingly patient-focused. Many developed countries have introduced at different times orphan legislation to incentivize development of orphan drugs. Contrary to general belief, being designated as an orphan drug does not automatically allow a regulatory authority to approve it more quickly or with less evidence than drugs intended for non-orphan populations. The standard for product approval is still based on satisfying the criteria for safety, quality, and efficacy. Given their statutory role as guardians of public health and patient safety, regulatory authorities understandably require a robust and comprehensive data set to be provided to reduce scientific uncertainty. Clinical development to prove safety and efficacy of a new product is most costly. It is also most challenging for new therapies intended to treat orphan diseases, given the limited pool of patients that the clinicians have alluded to earlier on. The problem from a resource perspective is the need to set up multiple trials to meet different regulations and requirements. The solution would be a common trial design, but this may not be possible. Although international authorities may establish so-called parallel scientific advice, such as between the European Medicines Agency and Food and Drug Administration, the respective agency do not have to arrive at the same view on the study design. The standard for approval of orphan drug is legally the same as the standard for approval of all other drugs in the United States, as I understand it. The FDA requires substantial evidence of effectiveness derived from adequate and well-controlled investigations. FDA has authority to apply the regulations flexibly and has done so in many cases, but there is no obligation for the agency to do so. In Europe, it adopts essentially the same regulatory standard. However, it is recognized by the EU legislature that in certain exceptional circumstances, a product license may be granted on the basis of less comprehensive data. In addition, for certain rare, life-threatening, and debilitating conditions, the EU legislature has created a regulatory pathway for a temporary license, so-called conditional approval, to be granted subject to annual renewal based on reassessment of benefit-risk assessment. The temporary conditional approval accepts that there is uncertainty of the submitted data, but recognizes also the immediate access to the product in view of an unmet medical need is sufficient to justify its approval. The uncertainty of the scientific data set will be addressed post-approval to generate more comprehensive data. A delegated regulation has been adopted by the European Commission to give greater clarity of the situation in which post-approval efficacy may be required. the legislative directions encourage greater use of alternative clinical trial design and post-approval real-world evidence. It is my understanding that U.S. Congress has legislative power to provide clearer directions to FDA to fully embrace less conventional and/or less commonly seen methodological approaches to establish clinical safety and efficacy in exceptional circumstances so that a new therapy is not unjustifiably denied or delayed. The regulatory risks and scientific uncertainties can be managed through specific enforceable post-approval safeguards or commitments to monitor safety and efficacy. This flexible and pragmatic approach may serve the public health imperative of improving patient care in a setting where there is a demonstrable unmet medical need. It has been said in various published literatures that when a treatment method fails, researchers must be clear that there is a true lack of biological effect, rather than failure due to inadequate study design. Therefore, the approval process ought to take full account of the detailed knowledge of the broader sources of information to help determine what is the right level of evidence to support safety and efficacy of a new therapy. In this context, Mr. Chairman and Senators, consideration should be given to the following points. First, what constitutes an adequate level of scientific evidence to presume strongly a favorable benefit-risk balance to support product approval? Secondly, is it feasible or practical to generate comprehensive data within a reasonable time frame following approval? Thirdly, can the scientific uncertainty of the submitted dataset be resolved by specific and enforceable post-authorization studies, including real-world evidence? Clinical development, as my colleagues on the right have already said repeatedly, clinical development is increasingly globalized. Greater cooperation amongst various national and regional regulatory authorities to agree on the design of multiple center clinical trials will greatly facilitate the efficient execution of product development in this particular rare disease to serve the patients with rare diseases and to optimize their care and management. We can strive to obtain the perfect data set, but the patients cannot wait. It is possible with the international cooperation to rely upon one well-designed clinical study to elucidate the true treatment effects of a transformative method of treatment. Mr. Chairman, Senators, I thank you again for your attention. I'm very happy to take any questions.
Thank you, Dr. Sang. Our last witness is Mallory Factor, who is a professor at Oxford and the founder and CEO of IntraBio, Inc., which was founded for the purpose of developing novel therapies for rare diseases.
Thank you, Senator Paul, and thank you also, Senator Casey, for participating in this, as well as the other senators. My colleagues to my right, I think, have covered a great deal of material already. I'm here today to share with you some observations on some of the obstacles that delay and even restrict novel orphan therapies from getting to patients, and some of the ideas how orphan drug developers and the Food and Drug Administration, the FDA, could collaborate more closely to bring treatments for rare genetic diseases to the point of approval so that they're available to patients with conditions or diseases for which there are high unmet medical needs. Rather than go over some of the same material that my colleagues have done, what I'm going to do is I'm going to give you a quick case study, based on the actual compound you saw being used there. IntraBio is a small biopharmaceutical company whose mission fundamentally is to advance patient interest and to develop novel therapies to treat fatal, rare, rapidly progressing genetic diseases with high, if not totally unmet medical needs. Company is developing a compound, N-acetyl-L-leucine, which is supported by both animal studies and numerous compassionate use studies in patients to be a potential treatment for rare disorders like inherited cerebellar ataxia, e.g. ataxia telangiectasia, spinocerebellar ataxia, ataxia with ocular motor, apraxia, Tay-Sachs disease, which there's other videos which I'm sure can be distributed, and Niemann-Pick type C, which you just saw, as well as common neurodegenerative diseases like Lewy body dementia, a form of Alzheimer's, and Parkinson's disease. Given the extreme medical need, IntraBio is prioritizing the development of N-acetylleucine for the treatment of rare genetic diseases, 3 in particular: Tay-Sachs, Niemann-Pick C, which you saw the video of, and inherited cerebellar ataxia subtypes. And these predominantly affect pediatric patients and are fatal, and they're rapidly progressive, and display a huge range of debilitating neurological as well as physical symptoms. And they have no treatments medically available. IntraBio has commissioned further safety pharmacological studies to characterize the safety profile and further non-clinical studies to investigate the optimal form and the mode of administration for patients. I'm just going to I have a lot of this material in here, and rather than just read from it, I'll talk to you extemporaneously. Hundreds of millions of doses have been used of this drug since 1957. Hundreds of millions. Tens of millions of people have been on this drug. Its safety profile has been shown to be safer than aspirin, and yet we can't even begin to put it into people here yet in a trial. I can go into great detail in the interactions with the FDA. The FDA is first and foremost concerned with risk. What they need to be concerned with is risk, benefit, and timeliness. And they do not understand the risk-benefit analysis. I'm not sure what we can do, but the real problem is try to reach somebody at the FDA. You can't. It is impossible to sit down other than in a formal setting with months. It took us 8 months to get a meeting. And I won't tell you how many children passed away from these diseases during those 8 months just to begin to have a discussion with them. And you have to submit we submitted a 150-page briefing book, which I'm not sure anybody really read thoroughly. What we need is the ability to sit down with the FDA, and these are good people at the FDA, but we need to be able to sit down with them and have simple conversations. There's a disease called GM1, which we've shown efficacy with our drug. We can't do a trial because there's 16 people right now alive in the United States. You couldn't get statistical significance. So what are we going to do with it? What are we going to do? Nothing. We can't. But we'd love to be able to sit down and say, hey, how do we help these people, since we are aware of these people using it unlicensed? Some of the people are even forced to buy chemical grade. Our goal is very simplistic. We would like to work with the FDA and the regulators to move forward on a timely manner and look at the risk-benefit analysis. And that's what I think is vital. The acts, the Orphan Drug Acts and things like this are terrific, but they help after these trials. They don't help, you don't get into the trials. They're after you get your NDA, your drug approval. That's where the benefit comes in, not getting it to the people. I thank you, and again, the script will give you a lot more information than I've just given you.
Thank you, and thank you to the entire panel, and I'd like to recognize Senator Casey for any questions.
I'll be very brief. You were also compelling, and your testimony was so captivating that I stayed longer than I had planned, and I'm grateful I did that and grateful that you're here. I'll submit written questions because they are written, and with the aid of capable staff, they will be much more articulate than I could muster today. But we're grateful for the work, and each one of you presented, I think, a challenge to us on these issues, and we're grateful you took the time to be here and to provide both your experience, your scholarship, and your passion. And we heard that throughout the testimony. Thanks very much.
And Senator
you, Chairman
Casey,
Paul.
thank you. And hopefully, maybe we could talk some more about any updates to the Orphan Drug Act. That we've heard some things maybe from Europe and other places that are working better. Maybe you and I could work together and see if there's something we could do to streamline, to even do a better job. Sounds like there has been some benefits, but maybe there's something else we could do.
So it's on the record, yes.
All right, thank you. All right. Well, I think we have had a good discussion. I want to go into it just a little more and then also encourage everyone on the panel and everyone who's interested in this, Mr. Dant and your group and other groups like yours that exist, to come together and let's try to have an organization that brings together these suggestions for how we'd update the Orphan Drug Act. It sounds like there were some successes. Senator Casey mentioned that there were 15 drugs approved before the act, and then all of a sudden there's 600. But I think science is developing so rapidly, and we mentioned DNA sequencing and the individuality of treatment, and that there may be a disease that only one person in the world has. You can't really have a double-blind clinical study for one person. You can't have it for 15 people. I guess one of my questions, this might be for the scientists, is the idea that you have to have a double-blind, randomly controlled study. Is that stopping a lot of what goes on with the Orphan Disease Act, or does the Orphan Disease Act actually understand and already make some accommodations for the fact that the numbers aren't big enough? We'll start with Dr. Strupp.
So I totally agree. This is a very big issue. Personally, I have designed 8 investigator-initiated trials. I'm very much involved in the design and also the statistical analysis. And the key number is that you do a sample size calculation. And doing a proper sample size calculation requires that you first have to estimate, was it a clinically meaningful difference? So that's one of the parameters. Then you have to add the power of the study and the alpha value. And then you often end up with sample sizes of 50 or 100 or 200 patients. You can do that.
That is then being accepted by the FDA with the understanding that you can't do 1,000 people?
Of course, 200 would be accepted, but if you have only 50 in the US, you cannot do a proper statistical design and analysis in the conventional way. And Mark Peterson pointed out in his statement what are potential alternatives for such analysis based on up-to-date, sophisticated statistics.
But given the current situation, you're saying currently it is not working. It is difficult to get the FDA to approve studies for orphan diseases, yes?
Yes.
Okay, Dr. Patterson?
Senator, thank you. I think just to come back to your point, the Orphan Drug Act, as you know, initially really incentivized companies to do studies, but it didn't direct the FDA as to how these studies should be conducted in rare diseases. The Cure America Act, I think, was terrific. It was very encouraging in terms of asking the FDA to explore adaptive trials, for example, and Bayesian approaches, but it didn't say, you shall do this. And I think that would be very important to give regulators those tools to say, you've explored this, you've shown it's appropriate. There's quite a literature that shows that these trial designs may be effective.
Right, and we've had some of this debate in the past, like on using European studies or Asian studies. And, we had the words to put into the bill that said the FDA shall do it actually or must do it. And everybody said, oh, no, we say that they can. So that people soften the words because they say, oh, we don't want to tell the FDA how to do their job. But at the same time, maybe sometimes we need to be more directive in the language because I think given the choice, often the status quo persists.
Senator, I would agree with you completely. Thank you.
Senator, I would also add that patients across the nation are jumping as fast as they can to get into the line to enter a trial. The negative is the trial designs are sometimes so narrow that many do not qualify because the clinical endpoints are so very precise, even drugs during trial that seem to help. And the parents will tell you, yes, my child was speaking afterwards, and now they're not, and yet the trial fails. Right. Trial designs of all-comer trials where because of the heterogeneity of the disease, 3 siblings can sit next to each other, but they present completely differently. The drug may help one, but not the other 2. We should look at all comers. And trial designs like the multi-domain responder index, which really takes the all comers, looks how they can succeed and how they.
And I think that might be something that you could help us with, particularly those who design trials. We talked about trial design being one of the things. So we have random double-blind studies as this gold standard that's been out there, and we have 1,000 people have to be, and it has to be half get it, half don't get it. So that's not appropriate for terminal illnesses, small illnesses. But if you could help us, not just today, but as we go through this process of if we're going to talk to the FDA or legislate how these designs, because it still has to have some latitude, you know, as to how the designs are, but maybe we have a standard now for design, and maybe you can help us with defining what the new design would be to allow the process to work better. Dr. Strupp?
May I add to that? You saw this girl, and if you just measure the time, that may be misleading, because if coordination improves, they will often take longer time because they do it more precisely. And based on that observation, the FDA was very open-minded when we suggested a so-called clinical global impression of change. That means experts evaluate the changes having a look at videos. They are blinded, on off the drug.
I think even a layperson might be able to discover that that drug seems to be helping. But the only thing I would say is the only thing you'd have to convince me is that it's not random. So you'd have to have enough videos to show me that this little girl isn't better some days and worse some days without drugs. And if there is no random improvement like that, and you have that dramatic improvement, it shouldn't take us 10 minutes to figure out that there's a great deal of benefit there.
Right, and then you don't need 50 patients. If I present you a pig which is fluent in Spanish, it does not to speak additionally Chinese.
Right.
So that's
This
very
drug
convincing.
was also the drug that's been treated for decades in France, and so it's not something that is poison. It's not something that people are dying from. There's no safety problem.
You mentioned a very, very important point, and we always have very vivid discussion with the FDA in terms of teratogeneity and cancerogeneity. Imagine most of these diseases affect children. So teratogeneity is unfortunately not an issue because they won't have children. So why do we have to do animal studies in 2 animal models if we treat children at the age of 13? And the same is for cancerogeneity. If you have a life-threatening disease with a significantly reduced life expectancy, and we have
sounds like you need somebody that's able to short-step this, so it's able to look at this, and maybe like what Dr. Sang was saying in Europe, that you give a temporary approval much quicker. But what you need is something that a CEO in a company will do, but the government is very, very hard to develop things that are adaptable. The government's very, very rigid. And I know the FDA wants the best. They don't want, you know, a horrible occurrence like we had, you know, with some of the things that were approved too quickly. But at the same time, I think maybe the pendulum is over here and it needs to come back towards the middle, and you need to give someone the prerogative to actually look at a video like this and say, my goodness, there's 16 people with this disease. Why don't we start more aggressively getting this to the people and still continue to study it and maybe have a temporary and have a review each year for safety and, you know, because, but I can't, we need to do something. I don't think we're doing enough.
I think we would even have a more impressive video with Tay-Sachs. I don't know if you have it in the files.
We can show it if someone can find it. Let's go to Professor Factor. If someone wants to queue up whoever's in charge of the video, if they can find the Tay-Sachs, we'll look at that. All right, go ahead. You had a point you had like to make?
The key, really, from our perspective, is early and frequent interactions with the FDA to make the development programs before the program begins, even before you do your filings, that you have a working relationship with the FDA. And what the FDA has to recognize more than anything else is the endpoint should be quality of life. It should, and the patient and caregivers really know those endpoints better than anybody else. Quality of life should be the key. And they really need to have a separate track for fatal orphan drugs with high unmet medical needs. And there has to be a separate track, which they do not have.
Right.
I can give you, at some point, I hope I have the opportunity to, an example of how it took us 8 months just to get a meeting.
Well, I think all of those things are important. What we need to do is just take them and turn this into something constructive and take the problems we are having and let's look at the law specifically and see how we can change the law. And like I say, some of this is we need the input of scientists on how to look at the parameters of what the government is saying we need for design to say how can we adapt that. And I think it's a point well taken that the Orphan Disease Act was mostly about encouraging companies to invest in this, but didn't necessarily make the FDA process easier. Dr. Sang?
Thank you, Chairman. I think that my colleagues, what they have been saying is that the requirements must be applied in a proper clinical context. That is the key test, so that we are not applying the requirements disproportionately. Very often, it's very convenient for, as a former regulator, very convenient for regulators to say that I need to tick all the boxes and do it mechanically. But very often, the law actually provides, my understanding of the US legislation is no different from other jurisdictions. There is flexibility. The question here is whether or not regulators are prepared to exercise flexibility to be a bit more bolder, applying the requirements proportionately. That is a key test.
Right.
Senator, thank you. I would add one thing, jumping on that as well, is there is great science already available that goes to what is referred to often as the valley of death, where because it takes so long to move great science through the pipeline to the FDA and then to treatment, it costs too much. So companies cannot invest in science to move it along, and so the therapies don't come forward. That's the value of the Open Act and the value of repurposing drugs and how we can get those companies to jump into safe drugs that already are proven safe that may help treat.
And see, this is the problem that I've been carping on for a while. There's idiopathic pulmonary fibrosis, and my political director's sister has it, and the drug she uses was, fortunately she lived in New York City and had a family that had the wherewithal to get her in a trial. So, you know, having some means, you don't necessarily have to be rich, but you got to have the means to get to a center, and then a family that was able to get her into a trial. But the drug had been legal in Japan for a decade. And the problem I have is that we're so rigid that, let's say there's a blood pressure drug, maybe 15 million people are taking it. And so your safety's been, 15 million people in France have been taking a blood pressure drug for 15 years. We're going to start over here with all of the safety trials instead of having a great deal that the preponderance of evidence is that we should really shortcut these things. And I think wanting to have zero tolerance, and we don't want to approve drugs that either don't work or are unsafe, but I think we have too much rigidity. And as Dr. Sang was saying, maybe some of it is getting people who are more open-minded to that. And we'll talk with the FDA director as well, who's a medical doctor, and see if we can talk some about the human element of being less rigid. But also, I think something needs to be done to the Orphan Drug Act to actually maybe separate a category or have a separate pathway just dedicated for the orphan drugs. But it's not going to be just orphan drugs. There's going to be, I think people's individual cancer is already being treated in an individual way. We've had testimony from people with ALS or Lou Gehrig's disease about how there's different varieties of it, and you're going to discover and have a treatment probably for one person or 10 people, that are a variety of that. So we just have to figure out how to get beyond that rigidity. Are we able to watch another video? Do you want to introduce it, Dr. Strupp? Put your microphone on.
So this was before treatment. It's an 8-year-old patient with a genetically proven Tay-Sachs disease. So without treatment, he could hardly stand unaided. And now click on the other one on the football field. So that was 7 weeks later. So I think this is...
You're convinced that this isn't random? There's not that kind of random...
He was off, on, and off, and colleagues who saw him couldn't believe how much he improved. He did not support our football team, but nevertheless, he improved very much. And go to the next, please. Not to the video, to the next slide, please. And click on the animal on the left and the right. So these are the complementary studies which are doing. And the left one, please, also. And this is a nice thing. This animal gets the same drug and the same dosage per kilogram. And you see the major difference. And again, if you will measure the time it takes till the animal reaches the bottom, the non-treated will be faster before it falls down. So we just have to have a global impression.
And so these studies are being done in Germany.
No, this is done in collaboration with Oxford, Francis Platt from the Department of Pharmacology.
And so, but then my question is, if you want to show these studies, what's the odds that the FDA is going to look at these studies that you've already performed or make you do them over?
So they liked the videos very much. July the 17th.
But are they going to be acceptable part of your presentation, you think, to the FDA?
Yes, yes.
Okay, good. Dr. Patterson?
Senator, I think just to comment, you very astutely mentioned the importance of deciding whether a change is random or not. I think this is a beautiful example of where you can use an N of 1 trial design, where you can blind, in fact, both the patient and the observer,
Whether
who
they've
can
had the
be
drug.
reviewing a videotape, so they don't know if the patient's on the placebo or the control. You can do that with appropriate trial designs, multiple N of 1, and get the information you need, which I think is still
Right,
scientifically
and
valid.
show the reproductivity. And this is some of the difficulty, like in Alzheimer's, does go up and down, and the cognitive studies are sometimes difficult to prove one or the other because people have really good days, and if you've had a parent or a grandparent with this, have a really good day and then have a bad, So once you can eliminate the randomness out of this, though, it shouldn't be that much more difficult, particularly and we should take into account the fact that the drug has been used for a long time in France by lots of people. Mr. Dant?
Senator, I would also add that in rare disease, natural history studies simply are not there because the diseases are so rare, which speaks to Dr. Patterson's point about the importance of patient-owned registries. Our partners in industry oftentimes have their own registries, but they're silos of information that are not shared.
So what goes into a registry?
What the parent, what the individual with the disorder sees, feels, does, they enter it themselves, and it becomes a living document that can be reviewed by physicians, by scientists, and by industry. We need the FDA to also look at those registries and understand that there is value in patient-reported outcome. The power of the patient in moving studies forward cannot be understated.
Okay, we're in the middle of a vote, so we're going to wrap up pretty quickly, and we've been kind of doing an open forum, and I kind of like this, so sometimes you get more from asking people what they want to say than asking them a specific question. So I'm going to open it one more time, and we'll see if anybody has something that would add to the hearing as we wind up here. I know I'm going to have to ask a question if you don't come up with anything. Professor Factor.
I'll add something very quickly. The particular drug that we're talking about that's used in France contains gluten, things like this in it. We're just trying to use the pure form, which causes all sorts of other problems by just taking the pure form, the active part of it. It's called a racemate, and the active form is is only part of it. But to get rid of the gluten and some of the other stuff that people react to, that is a whole bunch of other new tests, which makes no sense.
What I'd like to do, and we're going to wrap up here because I have to go over to the Senate floor and vote, is that I want to thank you all for testifying, for coming and taking your time. You don't get rich to be a congressional witness. You're not paid. In fact, many of you probably paid your own way here as well, and we really appreciate that because you believe in what you're doing. I really want to get a result. Just hearing testimony, we can do that, and if nothing happens, then I'm disappointed. So if you have further remarks or anything that you'd like to submit in writing, if there are other groups like yours, Mr. Dant, that would also like to submit something in writing, we'll accept that as well. And then let's continue the collaboration to try to see if we can do an update of the Orphan Drug Act that either has a separate tract, different design perhaps. And it's still my belief that whether it's an orphan drug or any drug, we live in a world, look, think Dr. Strupp's from Germany, but we don't say, oh, we're not going to listen to him because he's German. We have great doctors in Asia. We have doctors all over the world. And all of these studies, the world, the modern civilized world is an enormous world of medicine and science now. And we're crazy to stop it at the border and say, oh, now you have to Americanize all of the studies. And I think the scientific community already is very international. All the meetings are international. The studies are taken and written about in international journals. It's just the governments have decided to segment it off. And maybe, I hate to say it, but maybe even the EU could tell us something about how we should do this better. But, we would like to hear from you, and I hope the collaboration will continue. But thank you very much for your testimony.